Hey guys, today we're going to talk about Chapter 5, Microbial Nutrition and Growth. Now, microbes are found in even the harshest environments on Earth, deep in the ocean, and even recent studies have found microbes that live 100 meters under the deepest parts of the ocean floor. There's microbes that live in volcanic vents, in polar regions, and astrobiologists, when they're searching for life on other planets, are looking for evidence of microbes and microbial growth. Each species has limited environmental ranges in which it can grow, however. So specific temperature ranges, like some bacteria can survive in boiling hot springs, but can't be cultivated at room temperature in the lab. Nutrient requirements and host dependency can also differ between species. Microbial growth we'll define here as an increase in the population size of microbes. Reproduction results in growth. And remember, bacteria reproduce by binary fission, where one bacterial cell divides into two daughter cells. So we double the population every time cells reproduce. The result of microbial growth is discrete colony growth. So a colony on a bacterial plate in the lab is an aggregation of cells all arising from a single parent cell. Generation time, or doubling time, is the time required for a cell to grow and divide, and it depends on the chemical and physical conditions. So for E. coli under ideal conditions, it can double every 20 minutes. If the conditions are not ideal, say it's too cold, then E. coli may take as long as 10 hours to double. Mycobacterium tuberculosis can double every 16 hours under ideal conditions, or it can take 48 hours or more if the conditions are not quite right. For example, let's say 10 cells of foodborne illness pathogen in potato salad are sitting for 4 hours in a warm room. They're capable of multiplying to more than 40,000 cells. All right, and here's an example of a typical microbial growth curve. You've got time on the x-axis and the number of live cells, or the log of them, on the y-axis. So the higher the red line goes, the greater the number of bacteria. So when bacteria are first introduced into an environment, whether it's potato salad or your TSA plate, there's a lag phase. It takes a little time for the bacteria to start growing at first. They've got to find their footing and start from scratch, growing a new population. Well, once they get cooking, they experience a log phase, or a near exponential growth. And that continues for a short time, really. In a very short time, the bacterial population size will grow dramatically. But then it'll taper off and it goes into a stationary phase. You could think of this as reaching the carrying capacity. That's the most bacteria that environment can support, until eventually they begin to decline. So that's the death phase at the end there. And this happens when resources become scarce. Maybe they've used up all of the food resource in that environment. They may become overcrowded, and sometimes they'll even modify the environment so that it's no longer hospitable for them, right? Like the production and buildup of acid might make the pH in the environment such that it's no longer a good place for those organisms to live. But we see this same general growth curve pattern over and over in all different types of microbes and environments. So, nutrients required for growth. Organisms use a variety of nutrients for their energy needs and to build organic molecules for their own cell structures. The most common nutrients contain necessary elements like carbon, oxygen, and nitrogen. Other important elements are phosphorus, magnesium, potassium, iodine, and sulfur. There are also trace elements like iron, iodine, and zinc that need to be present. Now, carbon is the backbone element of macromolecules. And the four major macromolecules are carbohydrates, things like sugars and celluloses, fibers. Lipids are things like fats, oils, and waxes. Nucleic acids are DNA and RNA. And amino acids make up proteins. Nitrogen is contained in many organic compounds too. It makes up the nitrogenous bases of DNA, the amino acids in protein, and about 14% of the dry weight in microbial cells. It's often a growth-limiting nutrient. Growth ceases due to insufficient nitrogen sources for proteins and nucleotides. Nitrogen can be acquired by nitrogen fixation. Only some prokaryotes can do this, where they take atmospheric nitrogen and are able to fix it into ammonia or nitrates and nitrites that can be used. Nitrate reduction into ammonium is important here, and recycling nitrogen from the amino acids and nucleotides of previously living organisms, or decomposition. We need to make use of nitrogen that's already been fixed but is no longer being used. Oxygen is also important. Oxygen is a requirement for aerobes, so organisms that require oxygen. It also plays a role for obligate anaerobes, facultative anaerobes, aerotolerant anaerobes, and microaerophiles. All right, so let's have a closer look here at oxygen requirements for prokaryotes. On the left is an obligate aerobe. This organism can only grow if oxygen is present. They have an absolute requirement for oxygen. A facultative anaerobe, they grow better when oxygen's present, but they can grow without it. So we're kind of on a spectrum here. Obligate anaerobes cannot grow or multiply in the presence of oxygen. They need that oxygen to be gone and out of the way. A microaerophile has a very small oxygen requirement, but high concentrations of oxygen are inhibitory to their growth. And last but not least, aerotolerant bacteria are indifferent to oxygen. They can grow in its presence, but they don't use it to transform energy. And the special media that we use to determine these oxygen requirements is called fluid thioglycolate. All right, oxygen can have toxic effects, actually. So all organisms produce superoxide, or O2 minus. Superoxide is toxic to cells. It steals electrons, and it has to be neutralized. Superoxide dismutase is the enzyme that forms hydrogen peroxide in cells. So if you have two of these superoxides plus 2H plus ions, you can form hydrogen peroxide and oxygen gas. Hydrogen peroxide is also toxic to cells, and it must be neutralized. So how does this happen? Well, we have an enzyme called catalase that breaks down hydrogen peroxide in your cells. And the chemical equation is this. 2 molecules of hydrogen peroxide break down to yield 2 molecules of water and 2 oxygen gas. Now, if you have hydrogen peroxide in your medicine cabinet at home, you may have noticed that it has an expiration date. This very chemical reaction is one that occurs at a very slow rate naturally. So over time, that bottle of hydrogen peroxide in your bathroom cupboard will break down into plain Jane water and oxygen gas. But it takes years. The enzyme catalase can make it happen in seconds, which is incredible. And that's what makes it bubble when you pour hydrogen peroxide on a cut or a scraped knee or something. The bubbles you see are the oxygen gas in the water. All right, obligate anaerobes lack this superoxide dismutase. They lack catalase, and therefore oxygen's toxic to them and they can't survive. They don't have the biochemical equipment to deal with it. Water. All microorganisms do require water for growth. Certain environments may contain substances such as salt that interact with water molecules, which make water unavailable. So, if the solute concentration is higher in the medium than the cell, what would happen? What would happen if you put a cell in salt water? Solute is the dissolved substance like salt or sugar. Water would diffuse out of the cell due to osmosis. So cell cytoplasm would dehydrate and shrink. Water's always going to move to the area of the lower water concentration. So here you can see in the picture on the bottom left, we have cytoplasmic membrane is the green line. The cell wall is the purple, right? And the red dots are the dissolved substance, the solute, so in this case salt. And we can see water is this peach volume here. So if there's more solute outside of the cell, and that also means less water, right? Lower water concentration, then that means that water will move from inside the cell to outside the cell. And that'll leave you with this shrunken plasmolized cell here. The cell wall doesn't move, but the cell membrane will shrink up. This is one reason why a lot of foods used to be preserved with high concentrations of salt or sugar. It makes it very difficult for microbes to grow and spoil those foods if the solute concentrations are so high. Halophilos, however, love salt. There are some organisms that are obligate halophilos. They actually require high salt concentrations to grow. Facultative halophiles can tolerate high salt concentrations. So, for example, Staphylococcus aureus is a salt-tolerant organism which colonizes human skin. It's also an opportunistic pathogen. All right, some other important growth requirements are physical ones like temperature. And the effect of temperature on proteins is important here. Proteins denature if they get too high. So think of an egg, right? An egg is mostly protein. If that protein gets too hot, it will denature, right? And you have a fried egg or a hard-boiled egg. If you cool it back down, will that egg go back to being a liquid protein? No, it's an irreversible change. So protein denaturation is serious, and it's irreversible, and it can be deadly for most bacteria, which is why boiling or cooking our food is so important, right? What about cold on protein? If you were to freeze a raw egg and then thaw it back out, would it go back to being a liquid egg? Yes. Good. So cold doesn't denature, but heat can. All right. The effect of temperature on membranes can also be remarkable. So if the temperature is too low, membranes become rigid and fragile. Remember, membranes are made of those phospholipids, and they need to be fluid to function. If we get them too cold, then they won't be able to work as they should. If they get too high, the membranes become too fluid, too loosey-goosey, and that's not good either. All right, so have a look at this graph here. We've got temperature on the x-axis and growth rate of bacteria on the y. So there is a temperature where bacterial growth gets faster and faster and faster, right? Up to an optimum growth rate. But if you get too far beyond that, it plummets, right? So we know that somewhere around here is where denaturation occurs of the proteins, an irreversible change. These bacteria will never grow again. They're done. But an optimum temperature for bacterial growth is a very real thing. This is why our body temperatures and the body temperatures of most living things like to stay so constant. So human body temperature is right around 37 degrees Celsius. And most of like all of our normal microbiota, their optimum temperature is just about 37 degrees Celsius. Now there's a narrow range on either side of that which we can tolerate. But this is one of the reasons our bodies have fevers, right? We try and denature and cook out the pathogens faster than it will harm our own cells. All right. And you can look down here and see the plates that go with them. At 22 degrees Celsius, there's very little bacterial growth. At 30, we see a lot more. But at 37 degrees Celsius is where we see the most bacterial growth. All right. Organisms can also be classified by their temperature requirements. Psychrophiles are cold-loving microbes. These are some of the microbes that are responsible for like spoiling milk in your refrigerator. We also have mesophiles. These are moderate temperature loving microbes. So these would be the organisms that can grow on our bodies. Thermophiles are heat-loving microbes. And then there are some hyperthermophiles. These guys require temperatures of 70 Celsius or hotter. And here's a graph showing the temperature ranges of these types of organisms. And this is an example of psychrophiles growing on snow in the polar regions. And give it that reddish color. All right, another physical requirement of microbes is pH. Most bacteria grow between pH 6.5 and 7.5, so neutral pHs. These are the neutrophiles. Alkalophiles prefer pHs above 8.5, and acidophiles prefer pHs below 5.5. Very few can grow below pH 4.0, which is another reason why acid is used as a preservative for food. So foods like sauerkraut, pickles, and cheeses are often preserved from spoilage by acids produced during fermentation. There are microorganisms that grow in an environment with pH less than 1. That's incredible. All right. Thank you guys for listening. If you have questions, make sure you bring them to office hours or lab when I see you next. All right.